Welcome, everybody. My name is Shayonika Mohanta, Market Segment Manager for Methylation and Epigenetics at Oxford Nanopore. I'm delighted to welcome you all today to this knowledge exchange, where we will be looking at how to unlock disease mechanisms with multiomic nanopore sequencing. After the talks, we're going to have a live Q&A session, so please make sure to add your questions to the Q&A box, which you'll find at the bottom of your screen. You can also upvote questions that you want most answered, and we'll try to get through as many as we can. This knowledge exchange is being recorded, and it'll be made available on demand after the broadcast. Today, I'll kick off the session with a brief introduction on nanopore sequencing. I'll discuss the advantages of using nanopore for multiomic studies, including its ability to capture full-length DNA and RNA sequences, detecting base modifications directly, and delivering real-time data. These features allow researchers to uncover intricate disease mechanisms that might be missed with traditional methods. Our agenda also includes Scott Hickey, the Director of Genomic Applications, who will present how genomic and whole genome and single-cell transcriptomic sequencing helps uncover insights into personalized immune responses. Then Art Rand, a research scientist in machine learning, will discuss how nanopore sequencing examines methylation and chromatin structure in normal and malignant hematopoiesis. So taking a step back, at the heart of biology lies the central dogma. DNA stores genetic blueprint, the RNA transcribes this information, and then proteins carry out the functions of life. So to fully understand the complex biological systems or uncover the mechanisms of diseases, we need tools that can analyze these different layers of biological information. But here's the problem. Legacy multiomic technologies can be complex and cost prohibitive. In genomics, traditional methods like SBS, microarrays, and bisulfite sequencing are often expensive, they require complex workflows, and still fail to capture critical details like structural variants or methylation. Similarly, in transcriptomics, techniques like RNA-seq are really piecemeal solutions that require multiple steps to reconstruct full-length transcripts, poly-A tails, or RNA modifications. Oxford Nanopore redefines this approach. ONT represents a new generation of DNA and RNA molecular sequencing technology. It's truly unique as it's the only platform that offers real-time analysis, delivering rapid insights in forms that scale from our pocket scale MinION to high throughput instruments for population scale projects like the PromethION. Our technology analyzes native DNA and RNA and can sequence molecules of any length, from very short fragments to ultra long reads, making Oxford Nanopore an incredibly versatile platform. And at the core of this innovation is the nanopore, a tiny biological pore embedded in a membrane. DNA or RNA molecules pass through this pore, generating electrical signals. And these signals, or squiggles, if you will, are distinct for each base, A, T, G, and C. And they can even allow us to differentiate between modifications, like a methylated C versus a non-methylated C. And this direct sensing capability is what makes nanopore sequencing so powerful. These unique biological properties position us as a comprehensive platform for multiomic studies. You can sequence a variety of nucleic acids at any length and simultaneously detect epigenetic modifications like 5-HMC or 6-MA in DNA, as well as M6A in RNA. This enables researchers to get a holistic view of the genome, epigenome, and transcriptome, all from a single platform. And this comprehensive view is so critical when studying complex biological systems or unlocking disease mechanisms because it lets us connect those dots between the multiple layers of biology. This brings us to a key question. How can we simplify the process of generating rich, comprehensive data while avoiding the inefficiencies of legacy methods? With traditional sequencing technologies, answering these biological questions often requires multiple steps. You need to prepare several libraries, use different sequencing platforms, and then navigate complex data integration steps. This approach is time-consuming, it's costly, and can introduce potential for errors or missed insights. Oxford Nanopore eliminates this complexity. As an example, here is our direct DNA sequencing. With just one sequencing library, one sequencing run, and a single streamlined analysis workflow, you can generate ultra-rich data. This includes everything from structural variation and single nucleotide variants to methylation and phasing, all in one platform. Nanopore sequencing isn't just simplifying workflows, it's unlocking a new level of detail and integration that legacy technologies really can't match. So let's take a closer look at this efficiency and how it translates into meaningful results for your research. One of the biggest challenges in genetic screening today is that traditional technologies often miss critical variants. While they can capture simple changes like single nucleotide variants or small indels, they struggle to detect larger structural variants, such as inversions or copy number variations. And these larger changes can have a profound impact on disease mechanisms, yet they're often overlooked. Adding to this complexity is methylation, a key epigenetic modification that influences gene expression and impacts all the variant classes. Without the ability to analyze both genetic and epigenetic changes together, our understanding of diseases remains incomplete. Nanopore sequencing addresses these gaps by uncovering more than 80% variation than traditional methods and integrates methylation data directly. This means we can identify those significant mechanisms that would otherwise go undetected. Thus, introducing our multiomics toolkit that delivers ultra-rich data from a single platform, streamlining the study of both DNA and RNA. Whether you're investigating chromatin conformation, structural variations, or phasing in DNA, or exploring single-cell gene expression, splice variation, and fusion transcripts in RNA, Nanopore sequencing offers a unified solution. And at the center of this toolkit is our flagship PromethION platform. This is a versatile device that scales to meet the demands of any project. The PromethION 24 offers unparalleled scalability, delivering up to 30x human genome coverage per flow cell. It has the ability to process up to 48 genomes per week, or as many as about 2,500 genomes per year. It's perfectly suited for projects ranging from individual studies to population scale analysis. You get real-time base calling with Q20 raw reads using our high accuracy or HAC workflow. This includes alignment, demultiplexing, and of course, detection of the DNA and RNA modifications, all as the sequencing progresses. It's also future ready. The PROM24 system supports standard formats like BAM and FASTQ for seamless integration into existing workflows. And upcoming features include onboard analysis with epitome and third-party integrations, making it even more versatile. So whether you're exploring genomes, transcriptomes, or epigenetic landscapes, the PromethION 24 system delivers unmatched throughput, real-time insights, and adaptability for any research need. So now you may be wondering, with all this throughput and real-time capability, just how accurate are these reads? From a single PromethION flow cell, it's possible to investigate the human genome at around 30x and get high-quality genomics and epigenetic variants in a single run. Over 99.7% SNV F1 scores across the whole genome. This is again at the 30x HAC base calling. Methylation calling at the single base level is over 99.5% accurate, which is actually more than bisulfite sequencing. SV calling is over 96%, and this is basically as good as it gets because it's limited by truth sets. And you even get megabase scale phasing. All of these accuracy numbers can be obtained with live base calling on our P24 device with the HAC base calling model. And to make human genetics streamlined and accessible, we also have several end-to-end protocols available from sample to data analysis. These workflows make advanced analysis accessible and efficient even to those with limited bioinformatics expertise. Our human variation workflow can detect SNVs, SVs, CNVs, and methylation in a single analysis while retaining full epigenetic context. It's a powerful tool for capturing a complete view of genetic and epigenetic variation. Results are rapidly summarized with epitome, providing clear reports and actionable data. For oncology, we have our tumor or tumor normal workflow, which excels in retaining all the methylation markers alongside genetic variants. This improves the classification, accuracy of tumor types, and the identification of driver mutations. When working with liquid biopsies, the cfDNA workflow supports both short and long cfDNA sequencing. This retains methylation data, provides insights into tissue of origin and epigenetic profiling within just 6 hours. And lastly, our single-cell workflow enables full-length transcriptomic sequencing for isoform reconstruction, gene fusions, and SNP detection. It generates around 80 million cell-assigned reads per flow cell and supports emerging technologies like GEMX and Visium's spatial mapping. Now I will hand it over to Scott Hickey, again, who's the Director of Genomic Applications, and he'll take us through his groundbreaking work combining whole genome and single-cell transcriptomic sequencing.
Thanks so much, Shayanika. Hi, my name is Scott Hickey. I'm Director of Commercial Applications, and today I'm going to talk to you about a project that we have related to antibody sequencing. And this is a multiomics project sequencing both at the single cell level to get the single cell B cell receptor transcripts, and then also a germline haplotype resolved assembly of the B cell receptor. So just a bit of background and study design. So antibodies are really complex structures. They have a heavy and a light chain, so 2 copies of each that are paired in the final polypeptide. Those heavy and light chains are transcribed from 3 different loci in the genome. And those loci are the immunoglobulins. Those are then rearranged in B cells and maturing B cells in a process called VDJ recombination. So these loci are really complicated. They have, you can see the V, D, and J, C segments. These are the alleles that are within the genome, and then they are very similar to each other. In maturing B cells, those are actually edited and recombined into just one of each gene. And then that is paired together and matured into the messenger RNA that is transcribed into the antibody sequence. So there's a lot of interest in sequencing these at the genome level to understand the diversity that is within an individual cell. And then these are also hypermutated and expressed when they're in the final B cells and making the antibodies. So we also wanted to look at the single cell expression of the transcripts in each individual B cell. So just a case study that we did from the same sample after an MMR vaccination. This was split into 2 workflows. So the top workflow is genomic DNA that we extracted from the non-B cells. So these are all the cells that do not have the rearrangement, the VDJ recombined. So that should have all of the alleles in the genome non-rearranged, and we can do our whole genome workflow and de novo assembly to get those annotated alleles. And then the bottom workflow is we isolate B cells from that same blood sample, and we do FACS sorting to isolate the cells that are expressing antibodies. And we can do our single cell sequencing workflow. We did this with 10x Genomics to get the expression profile. So this is a multiomics data set from the same sample where we can pair the expressed antibody sequences with the germline alleles. And there's a lot of downstream analysis that you can do when you have both of those analysis components. So starting with the top part, which is our genome assembly workflow. We did a haplotyped resolved germline assembly of the B cell receptor. We did this with the PromethION device. So we did a whole genome workflow using one PromethION flow cell. We generated about 120 gigs of worth of data. Mean coverage was about 45x, and that was enough to do an entire de novo assembly. The read length N50 was around 20 kb, and we had Q scores in the Q22 range from our simplex reads. We also had duplex reads from this data set up to Q31 in accuracy. And then we wrote a custom de novo assembly pipeline, which is available in our preprint referenced at the bottom of the slide. And this is using Fly as the assembler. And then we haplotyped with hapdupe. And essentially, we do a whole genome assembly, and then we pull out all the immunoglobulin sequences, the BCR sequences, and we can correct the errors using SV callers like sniffles, correct phase blocks, and get our final assembly. This workflow can also be done with our adaptive sampling workflow. So this is where we enrich sequence based on the software alone. So you can enrich directly on the device with using Minnow. You basically pass Minnow a bed file that has your regions of interest that you would like to enrich. And then when strands are being sequenced, they are sequenced in real time. If they align, so the base color and alignment is done in real time during sequencing. If the strand aligns to your reference file, to your region of interest, it is kept and continued sequencing. But if it does not align or it doesn't match the region of interest, it can be reversed and ejected out of the pore. So this is no additional library prep, but you can enrich for regions of interest in the genome. So for example, we did this with a panel of immune-related genes, and specifically the BCR immunoglobulin loci. So you can see IgK, IgH, and IgL, the heavy and light chain loci, are all in here. And then we also had 750 additional targets related to immunology and immune genes. So we can look at all the variants within these genes. You can see that using the adaptive sampling workflow, we get around 50x coverage average for all of these targets on a single flow cell. So that's really nice. And the Ig immunoglobulin loci were even higher than that. So we can take those into our assembly workflow as well. So after we enrich our immunoglobulins, for example, and we do our genome assembly, this is what it looks like. IgH, for example, is at the end of chromosome 14. And this we fully resolved into two haplotypes. You can see the purple genes, those are the V genes, the variable domain of the BCR. Those are all really highly similar sequences, and we can annotate those with the alleles using IMGT database. You can see that the haplotype 1 and haplotype 2 alleles are slightly different in certain areas. And then in the D genes as well, we see some D genes that are different between each haplotype. So this is maternal and paternal copies of the IgH assembly that we can fully annotate and resolve based on the haplotype and the VDJ C alleles that are present in the individual's genome. So then moving into our single cell transcriptomics workflow, after we have the assembly, then we wanted to actually look at the B cells and the single cell expression and the antibodies that are expressed in those cells. So a reminder, we took the B cells directly from that same sample. So we have a paired genome assembly from that sample already. And then we have the B cells that we're sorting, purifying, and then putting into our 10x single cell expression workflow. And we selected two different populations of B cells. So when B cells are activated based on antigen recognition and expanded, colonially expanded, or proliferated, they can differentiate into subpopulations of B cells. The two that we were interested in were the plasmablasts, which are highly antibody expressing and secreting. And so those have the highest number of antibody transcripts. And then we also took the memory B cells, which have the reservoir of all total antibodies that are expressed and circulated in the body. We then put these cells into the cells go through the 10x genomics device and workflow, and then we put that into our library prep workflow. So this is available on the website. The full protocol is available, but essentially it's really straightforward. You just take the cDNA that comes right out of the 10x genomics protocol and right off the device. You don't fragment it because you want the full length transcripts, and you amplify with specific primers, and then those go right into our ligation sequencing chemistry library prep. So there's an optional step to enrich for full-length transcripts. We support the 3' and the 5' in the Visium workflows currently from the 10x Genomics kits, but essentially take the cDNA directly into the Oxford Nanopore library prep kit and get the full-length transcriptomics reads out. Then in terms of data analysis, we do have a fully automated end-to-end workflow for your single cell data. This is called Workflow Single Cell. It's in our Epitome pipeline. And it takes the raw base called single cell reads and provides at the end your gene expression and isoform expression matrices. So it's doing barcode and UMI extraction. It's aligning to the reference transcriptome, and then it's correcting barcodes and UMIs and generating your outputs. So if you're familiar with Cell Ranger from 10x, this is similar. It works on Nanopore data, and it provides the similar QC reports, UMAPs to look at your single cell clustering. For example, so this is our memory B cells, and we can look at the clustering based on gene expression and also isoform expression. So if we just look at the gene expression, this is similar to what you would get with short reads. You have some clustering, but the advantage of having Nanopore full-length transcripts is that you can also look at isoform expression from the same data set. And so on the isoform expression, we do start to see more subpopulations of B cells that are expressing similar isoforms. So this is a really powerful way to get at underlying biology. These are all memory B cells, all the same types of cells, but we're starting to see subclustering of these very similar cells based on the isoform expression. And why is that? Well, we can look at differential isoform analysis now between the different clusters. So for example, we saw about 1,800 isoforms that were differentially expressed in this memory B cell population. We see BCL2 as an example where it has 3 different transcripts that were expressed in 2 of these clusters, and there we can quantify the amount of differential expression between these 2 clusters, hinting that one of these clusters is using the BCL transcript that the other one is downregulating, for example. Okay, and then we finally wanted to look specifically at the single cell antibody consensus sequences. So we took the workflow single cell output, so the reads directly from the first part of the workflow, and then we built a custom workflow specifically for antibody sequences. This is also available in the preprint. And essentially it's taking out all of the immunoglobulin, so heavy and light chain reads, it's grouping them, and then it's polishing them with Medaka to generate a consensus sequence. And then we're pairing the Ig heavy and Ig light chain sequences together per cell. So you get a single cell antibody consensus sequence that you can then use for downstream annotation using IG-BLAST, for example. So why do you want to do this? Well, the full-length antibody sequences give you additional information, for example, about class switching. So antibodies can be different, can have different classes, different isotypes. These are IgM, IgG, IgAs, for example. And then within the isotypes, there's actually subclasses of isotypes. And we know that the short read data sometimes is not able to resolve things like IgG1 versus IgG3 subtypes. And with the full length antibody sequence, consensus sequence, you can get the full constant region, and that informs what the downstream biology of these antibodies are. So the long reads help you to resolve not just the variable part of the antibody, but also the constant region. And this is important because then you can look across different populations of cells and see if there are isotype or class switch recombination events happening. So we looked at the B cells versus the plasmablasts and plasma cells, which is a third type of B cell subpopulation, and we can look at the isotype diversity within each class. Okay, so then now with the multiomic data sets that we have with the germline assembly of the immunoglobulin loci and also the paired heavy and light chain antibody consensus sequences, we can look at downstream analysis, complicated things like somatic hypermutation rates. So these are taking the antibody sequences, mapping them back to the personalized reference genome that we have and looking at how much frequency of mutations and how many mutations per cell compared to the reference allele that we sequenced in the assembly. So this is really powerful, and we can look at, we can stratify this by isotype again, because these are full-length sequences. We can then look at different mutation frequencies within the populations of B cells. For example, we see a rare IgD isotype of cells in the memory B cells that has a higher percentage of mutation frequency compared to the other cells, representing that those cells are more highly mutated compared to the reference alleles. Then we can also look at V gene usage. So this is looking at all of the annotated V genes and haplotyping those and stratifying them by isotype, so we can tell which haplotype is expressing which allele or which antibody. So this is really just a high resolution picture of what antibodies are being expressed from what germline alleles. And you can annotate these based on public databases like IMGT. However, we found that if we map these back and annotate them according to our personalized reference genome, we have a lower number of ambiguous calls. So the public databases have trouble sometimes discerning which V gene is being expressed because the V genes are very similar to each other. If you narrow that down based on what you know is in the personal reference genome and what alleles are present, then it gives you a higher accuracy assignment. Then finally, we looked at binding and neutralization of certain clones for these antibodies. So we took clones, we expressed them in transfected cell lines, and we can do ELISA, FACS binding assays, and then also neutralization and infection assays. So we found 2 clones from the ones that we expressed that show binding to the measles F protein. We find another clone that binds to mumps and rubella antigens. And then we found one that was fully neutralizing against the measles virus. So in a cell infection assay, it's actually protective against measles infection. So this is really exciting. It shows directly from sequencing data and just from consensus generation of antibody sequences, you can identify truly neutralizing antibodies that are actually protective against viral infection. Okay, so in summary, we took a multiomic approach for studying the immune system and the immune response. This was based on both the germline B cell receptor assembly and also pairing that with single cell B cell receptor data, antibody data. This was donor specific so that we can map the transcripts that we had back to the germline allele annotations. We can look at differential isoform expression because we have the full-length transcripts in different subpopulations of B cells. And then we can pair those heavy and light chain sequences, get a full-length antibody consensus sequence, and use that for downstream analysis, for example, V gene usage, somatic hypermutation rates, and validation of select clones against MMR antigens and binding and neutralization assays. So thank you so much. This was a collaboration with Dr. Ben-Hur Lee at Mount Sinai. So thank you to his team and also to the applications team that worked on this project, and happy to take any questions. I'm going to pass it over to Art Rand next, who's going to talk about methylation-dependent mechanisms with nanopore sequencing.
Thank you, Scott, for the introduction. So we're going to have a little bit of a shifting gears now. We're going to talk about methylation and how you can look at methylation and use methylation and chromatin accessibility to, study your biological system. So first, because this is a nanopore knowledge exchange, we have to talk about how the nanopore works and how you can actually detect methylation with it. I don't think this will be too surprising to a lot of people, but I just want to emphasize that in the animation in the center, you can see that a strand of DNA or RNA, but in most cases, in the case we're going to be talking about, it's DNA, is actually traversing through the sensor. And so if you have chemical modifications to that DNA, they actually are interacted with by the sensor. So it's not it's not a chemical treatment or something that's a proxy for there being a chemical modification or a methylation. It's the actual atoms themselves are flowing through the sensor. So just to bring everybody up, what are DNA methylation modification, or what are base modifications and chemical modifications? Most of the time, we're going to be talking about methylations to the nucleoside, and most of the time, we're going to be talking about methylation to cytosine. So that's on the left side of your screen here. Most of the time in mammals and in the study I'm going to talk about now, it's 5-MC and 5-HMC, so 5-methylcytosine, 5-hydroxymethylcytosine. And then we're also going to talk a little bit about 6-MA. You can also do, you can detect modifications to RNA. And I've just sort of put some of those that we provide on the right side there. So today we're going to be talking about how methylation and chromatin structure can be studied with the Oxford Nanopore platform. So just to bring everybody back up to speed on that. So in the eukaryotic cells, you have the nucleus, and the nucleus contains chromatin, which is broadly speaking organized or divided into two kinds of regimes. You have heterochromatin, which is tightly bound and usually not very transcriptionally active because DNA binding proteins have a hard time accessing the DNA structure itself. And then you have euchromatin, which is essentially the opposite, which is less tightly bound and more available to DNA binding proteins to go and find locations to do their job, and usually more transcriptionally active. So thinking about this in a hierarchical way, you have the nucleus on the left, and then you have these topologically associated domains where regions of DNA that might be very far away in the primary sequence can be brought into close proximity physically within the cell, and then zooming in just a little bit further, looking at some of the proteins that are involved in actually organizing that structure. So the case we're going to be talking about today is some work done by Dr. Aaron Vinnie at Columbia, where they look at myelodysplastic syndrome, and specifically the work we're going to be looking at is what happens when you take the STAG2 protein, which is one of the proteins in the cohesin complex, and you knock that out in bone marrow. So they have a mouse strain that has this protein knocked out. And we're going to be looking at what happens to the CTCF binding dynamics, so or the CTCF binding in general, which is this sort of kidney-shaped protein in this cartoon here, which is influenced by DNA methylation. So we're going to be looking at what happens when in DNA methylation at CTCF binding locations when you take this protein out. And this protein, as you can see in the cartoon, is responsible for forming some of these loop structures and stuff like that. So just as sort of a prelude to that, when you do knock out this protein, this STAG2, like I said, what ends up happening is we observe, and this is from chromatin immunoprecipitation sequencing, you get this relatively large gain in CTCF binding location. So in the Venn diagram on the right, in red, that's sort of the wild type case where you have some number of CTCF binding locations. And then when you knock out STAG2, you get this, relatively large, almost doubling of CTCF binding locations. So we're going to be looking and seeing like what's going on there. So just hold that in your mind for a moment. I'm going to quickly talk about how you actually do this, because if you're studying something, you're probably going to want to follow some of the same similar steps when you're performing your analysis. So it should be no surprise to folks that if you want to do methylation with Oxford Nanopore, it's relatively simple in that you can just sort of turn it on. It's the way I like to tell people is even if you're not detecting methylation, you are detecting methylation because it's just sort of part of how the whole system works. So, if you're using Minnow, you just sort of pick which methylation models you want to use. And then if you are doing base calling or some kind of post hoc analysis where you need to rebase call or redo methylation detection, you can do that on the command line with our program called Dorado. So the two programs that I'm going to basically talk about, mostly the second one is Dorado, which is the base calling program I just mentioned, and that runs in Minnow. And the other is ModKit, which is sort of a analysis suite that helps you pull methylation information out of your data and manipulate it. And the whole sort of ethos behind it is that it's trying to make you more efficient in figuring out what's going on with the methylation data and help you iterate quickly. So these two programs are relatively easy to get, especially if you have like a Linux computer or something like that, and they're well supported by Oxford Nanopore. So once you do base calling, or once you do your sequencing and you have your base calls, I call this kind of step 0. It's the first thing you probably want to do just to make sure your methylation information is sort of intact and what you expect, because this is a like 30-second test just to make sure that everything sort of looks the way you want it to look. And so the first command you probably want to do is this thing called modkit summary, where it's basically just going to skim some of your reads, and it's going to say, okay, I'm going to look at the probability distribution of the methylation calls, and what you really want to see is something that's kind of a histogram that I'm showing on the right here, where all of your methylation calls, the probability is sort of pushed to the right, which means that they're high confidence calls. If it's detecting methylation, it's pretty confident that it's detecting methylation. That's also reflected in this threshold number here. That number you want to be relatively high, like I've shown here. And then also the sort of fractions of methylation, they should more or less correspond to what you expect in your sample. Now, if you're doing something that's totally new and you don't know what to expect, obviously that's a different story, but I think most people probably have at least some ballpark idea of what they should expect to see. So that's kind of a good, just like, okay, I have what I think I should have. And then, as I mentioned, the probability distribution you can get from this other command. And then this is skipping ahead just a tiny bit, but you can convert your methylation calls into a big WIG format, which is usually the sort of preferred way to look at sort of continuous histogram-like data on the genome. So this is where if you're looking at a genome browser, you can make a track that has the 5MC calls as a function of the genomic position. So that's kind of handy. All right, so question is, like, can we start to see a phenotype in the methylation in these knockout cells? So as I mentioned before, you have this big gain of CTCF binding locations in the knockout. Let's start looking and seeing what's going on at both what we call ectopic sites, or these new CTCF binding locations, compared to the ones they have in common. So one thing you can do, ModKit has a function called entropy, which calculates something called DNA methylation entropy, which you can think of as a measure of the disorder of the methylation patterning in a region. So if, for example, you had all of your reads had the exact same methylation pattern, that would be very, very low entropy, versus if all of your reads have slightly different patterning, that would be higher methylation entropy. And so what you can see is this sort of like bimodal distribution of the ectopic sites having sort of higher methylation entropy, which is something that you might want to investigate, because that might lead you to think that there's more change and dynamics going on in those places, because all of your reads the methylation is not ironed out. It's sort of, each one's a little bit unique. The next thing you can do is the first step 1 after step 0, I suppose, is you do this thing called a pileup, where you're basically going to aggregate how many methylation calls you have at each genomic position. This will come out of epitome if you're using some of the epitome workflows. And then once you have this table, Second command I'm showing you here is called localize, which is a really powerful method, really powerful but simple method to just look at how methylation is changing at different places in your genome. So you could be doing, promoters or something like that. In this case, we're looking at these CTCF sites, but this command is like very fast to iterate on. And I like to tell people that this combined with sort of like bed tools, which is sort of a genome arithmetic program, lets you kind of say like, okay, I want to look at these sites, but take these other ones out, look at a pattern, internalize that in your head, maybe go and manipulate that a little bit or do like a, outer join or something, go do that again. And you start to find these patterns just by, iterating on them and looking at them. And so the idea is that this makes that easier to do. So to actually talk about the data for a moment, this is sort of the pattern you expect to see. So I'm showing the wild type on the sort of center left here and the knockout on the right. And you see this like big suppression or hypomethylation right at the dead center where the CTCF is binding. That sort of fits our model of how CTCF and methylation work. And this is at the low entropy sites for both the knockout and for the wild type. So this is kind of like the normal. This is the wild type phenotype, right? But if you start looking at these overlapping CTCF sites, and if you look at the ectopic sites in the knockout cells, you start to see that it's not quite as much of a hypomethylation suppression, and especially in the ectopic ones, you're getting almost 4 times more methylation at those places, even though CTCF is still binding. So there's something going on that's allowing CTCF to bind at these places that you would normally not expect it to, or something's happening that's kind of, fighting back the CTCF binding there due to the STAG2 being knocked out. So this is sort of like where we're starting to look next is like, well, what specific motifs maybe are at these locations? Are there specific CPGs that are important or, more predictive of CTCF binding or not binding? So this is sort of an area of ongoing research. So the next thing we're going to talk about is how you can do chromatin accessibility studies simultaneously with methylation detection using our platform. So this technique has been around for a little while, or techniques like it. They all sort of follow a similar design philosophy, which is what you're going to do is you're going to take chromatin, or you're going to take permeabilized nuclei containing chromatin. You're going to treat them with a methyltransferase. It's going to put an exogenous methylation in your sample. So exogenous meaning it doesn't normally occur in that biological system. And then you're going to simultaneously measure, the base call, the read sequence, the endogenous methylation, so this is the CpG methylation in our case, and then the exogenous methylation is going to be indicative of open chromatin. So in the case we're going to show here, you on the left side, you have your native chromatin. You use a methyltransferase. In this case, it's a non-specific 6-MA methyltransferase, which is going to find regions that are open, that the methyltransferase can essentially fit, and it's going to methylate the A residues into 6-MA residues. And those are, that's your signal. And so on the right, what I'm trying to show is that the advantage of this technique is that this happens and you still get your long reads that contain all of the other information. It's all sort of one assay. So if you had, for example, an allele that has methylated CpGs on the top here, that's accessible, right? So you can see that's accessible between these nucleosomes versus an unmethylated allele, which is inaccessible, you'd be able to see that. And you can actually just look at these reads on IGV often, and you can just, it like pops right out. So that's sort of the advantage of doing it this way. You can obviously also do your phasing and everything else that comes with, long reads, all of those advantages. Historically speaking, it was a bit of an issue that when you did this treatment, your accuracies suffered. So that's the density here, kind of towards the left, that when you did an a 6-MA exogenous treatment, you would see a regression in accuracy. We've worked pretty hard to essentially remove this drawback, that you can now get accuracies that are nearly equivalent to untreated, and I think we can actually bridge this gap even or close this gap even further. So there's really no disadvantage of doing this treatment, short of the fact that you do have to handle nuclei and stuff like that. But the data that come out are essentially equivalent. You just have this extra layer of information, extra layer of chromatin accessibility information along with it. So this is depending on when you're watching this, this might be released or it's something we're still developing a little bit, but you still can go find publicly accessible protocols. But when we do this and we use the modkit localized command like I was showing earlier, you can start to look and make sure you're seeing patterning you expect. So here what I'm showing on the left two panels are 6MA enrichment at various features. So this is at ATAC seq peaks. So that's another method for saying, or if we're looking at open chromatin, you see this enrichment right dead center of the ATAC peaks, which is great. For DNase hypersensitivity, that's sort of a similar idea. This is actually a really interesting one because you can actually see not only the enrichment at the DNase hypersensitivity sites, but you can also start to see the internucleosomal patterning. So you see this oscillation that is, you're seeing methyltransferase accessible regions between nucleosomes, which just goes to show how tightly regulated these structures are in the cell. And then finally, over on the far right, when you predict regions of open chromatin, so you take all your data and you say, okay, show me where the chromatin is accessible, you only use the 6MA signal, the 6MA information to make those predictions. And then you can plot that, and that's what this blue line is showing. It's actually very similar to DNase hypersensitivity, which is good. But then you can overlay the 5MC information because those reads also have 5MC information. And so when you do that, you see this hypomethylation, this sort of like dip in 5MC methylation around predicted regions or regions of predicted open chromatin, which is also another one of these, you get what you expect, good checks. So so this is sort of something you can do today with some open source tools. We're continuing to iterate on this and make it more easy to run and put them into epidemi workflows. So that's something that's coming. So with that, I'll wrap up. And I'd like to say a big thank you to Dr. Aaron Vinny at Columbia, who shared some of his work with me, and I shared it with you today. I hope I've shown you can do this kind of analysis relatively easily with our available tools, Dorado, Modkit, and Epidemi, and just to emphasize that I believe that it really helps if you can iterate quickly, look at data, make new hypotheses, look at more data, look at generate new hypotheses. That's what we try to do. So thank you.
Thank you so much, Art and Scott, for those excellent presentations. I just wanted to take a moment to briefly summarize what you've heard today on how Oxford Nanopore enables multiomic analysis. Our technology, as you've seen, allows for native DNA and RNA analysis, offering deep insights into not only genomics, but also epigenetics, as well as transcriptomics and single cell. One of the key advantages is, of course, our streamlined workflows that are designed to be accessible and efficient, even for those that have limited bioinformatics expertise. This means you can achieve advanced analysis without that steep learning curve. And when it comes to methylation and variant detection, we also have powerful tools. We have Minnow and Dorado that help detect both endogenous and exogenous methylation during sequencing, while Epidemi delivers intuitive reports for single nucleotide variants, structural variants, and transcriptomic data. Additionally, our Modkit gives researchers the flexibility to explore base modifications like 5MC, 5HMC, 6MA, etc. And of course, scalability is a huge priority for us, so our PromethION platform supports population scale throughput with onboard tools, making it a really versatile tool for a wide range of research applications. If you'd like to dive deeper into how our technology can support your research, you can find more information online. Okay, well, I'm afraid that's all the time we have today. Thank you so much for all of your questions, and thank you so much, Scott and Art, for joining us and for your presentations. You can find more information about today's broadcast using the QR codes on your screen. This knowledge exchange, along with all of our previous broadcasts, are available for you to watch online. So please visit the Oxford Nanopore website to check those out. Today's broadcast specifically should be available by next week, so just give us a few days. And you can also find more information about our upcoming webinars, knowledge exchanges, etc. on the website as well. Finally, we would really appreciate your feedback on how this knowledge exchange went. You can find a short survey on today's broadcast in the Q&A section at the bottom of your screen. You can just click on the polls tab. This will remain open once the knowledge exchange ends. If you do have any further questions about your projects or how to get started with nanopore sequencing, please do reach out to us. You can contact support@nanoporetech.com. Thank you all once again, and enjoy the rest of your day.